Hello, and thank you for joining us. I'm Kristen McPike, Program and Alliance Manager at the Antibody Society. Today's scientific briefing is one in a series designed to inform and educate our members and the broader scientific community about topics related to antibody discovery and development. Our expert speaker, Dr. Brady Wu from Biointron, will be discussing new trends in XDC and our experience with us today. Please note that this briefing is being recorded. You can add any and all questions to the Q&A box, and the questions will be answered at the end of the presentation. Finally, there will be a couple of poll questions towards the end of the presentation that we'd love your participation in. Without further ado, I'll turn the show over to our speaker, Dr. Wu.
Thank you, Christine. So the part will be divided to 3 parts, or a little bit mixed. I will mix the experience from Biointron and also with XDC's history and development. So let me go back to this story. So I think everyone will agree with me that today is the era of multi-specific therapeutics. And once the monoclonal antibodies came out in the 1980s and 90s, the field has been evolved into a multi-specific setting, including antibody-drug conjugates, siRNAs with GalNAc, and also antibody-cytokine fusion proteins. And even in the field of small molecule drugs, we have similar concepts, including PROTAC and also molecular glues. And of course, the great development of bispecific antibodies, including engagers and enzyme mimics, and also some other mechanisms. So one molecule will take different mechanisms in the body that is becoming an obvious trend in the industry. So today we are talking about antibody-drug conjugate. Antibody-drug conjugate was is composed by 3 different parts. First one is antibody, obviously. This is a guidance system for cytotoxic drugs. And the drug, you can see later, most of drugs are cytotoxic drugs. Those chemo drugs are not available individually, mostly, but when they are conjugated to the antibodies, I think they saw the rejuvenance of the field of chemo drugs. Of course, the last part and sometimes missed ones are the linkers part. The linker is quite important, and a lot of different mechanisms of linkers. So if you go back to the history of ADCs, the first ones are CD33 ADCs with hydrazone connectors. And of course, the great companies, Genetics, later named Seagen and then acquired by Pfizer, developed the mechanism with myelamine group and also interchain cysteine conjugate along with arostatin. That's a very well developed system in the ADC fields. And I want to point on to the great development by Daiichi, the DXT platform that further evolved the field of ADCs. They developed a payload with lower toxicity with similar setup, but on the same target and even the same antibody, but the therapeutic effects are totally different compared to the earlier version of ADCs. And you can see later there are a lot of different developments in the making of the ways of making of antibody-drug conjugate. Again, several, I think a little bit around dozens of products has been approved, but the field are converged, especially in the cytotoxic region. The field are converged to a few different type of payloads, including MMAEs DM1, DM4, and also XDCs. So when we are talking about generations of ADCs, when I went back to 10 years ago, we are talking about 3 different generations. And the first generation ADCs are called random conjugate ADCs, including 2 major ways. One is lysine, so it's a conjugation with amyloid. And in the antibodies, there are about 80 different lysines in the sequence, and probably 20 of them can be conjugated. So at the end, you have a mixture of the different DAR value products. So in the end, you will have a heterogeneous mixture of the products, and of course you have an average DAR value of the products, but if you consider 2 different DAR values, DAR 0 compared to DAR 8, they will have different hydrophobicity, developability, and also efficacies. So that will make the clinical more difficult to operate. Then it comes cysteine conjugation. When you first reduce the disulfide bonds on the antibody and then conjugate that by maleimide group. And if you go with DAR4, then you have again a mixture product, but several different DAR value products, because you only have 8 cysteines can be conjugated to the ADCs. And if you go higher, if you go DAR 8, you have 1 form of the final products. So that is where the DS8201 came in. It has a DAR value of 8 with DSD, and at the end you have one form of the final product, and it's a very pure product. So it contributes to the clinical effects of the molecule. Then we go to the evolution of the fields, and we talk about the random conjugation of lysines, but there are some other groups developing a peptide which will bind to the Fc region of the antibody, and then you put the group on the affinity peptide, and it will be transferred to the lysine, a particular lysine on the antibody. So in that way, you can make the conjugate site-specific. And also we have DAR4 molecule with transitional metal ion, and that will make the cysteine conjugation only on the cysteines on the light chain, between the light chain and heavy chain, not on the hinge region. And of course, we have the bridged conjugation. You conjugate 2 heads on 2 different cysteines, and that will make the linkage only on the hinge region. So in that way, you can make the cysteine conjugation even more site-specific. And we also have enzymatic conjugation. There are a lot of different ways. I just read a review published on Antibody-Therapeutics, but it was talking about dozens of different enzymatic conjugation. But I think the normally used ones are SOTIS, the tag LPETG. The SOTIS will transfer the payload on to the end of this tag, and also Q tag used by microbial transcriptions. But there is another residue will be available by this enzyme if you cut off the glycans, and the 295 position of the antibody glutamine will be exposed, and the microbial transcriptions can add the linker onto that glutamine. And of course, there are famously developed glycoremoduling conjugation with different setup 3 enzymes, 2 enzymes, and sequential conjugation methods. And then it comes the engineer cysteine conjugation. That is where you put a cysteine mutation on the surface of the antibody. Of course, you want to choose a site very carefully, and the site will not affect the structural stability and function of the antibody. And you can use some ways to conjugate the payload to that specific site. And the last one on this page is non-natural amino acids incorporation. This is a natural tyrosine, similar to natural tyrosine conjugation. No, sorry, it's a ketone bond, but you can use other ways such as natural tyrosine or the azide bond. So I want to show some very simple examples. We have been doing a lot of different conjugation ways. So this page is showing you the most simple ones. The DAR8 conjugation, very simple. And this is a little bit more difficult, but you might recognize it's a benchmark product very similar to immunogens. Yeah. Yes, immunogens products, and it's a lysine conjugation along with two-step reactions with disulfide bond. So the conjugation is quite difficult, but we can manage to do that. And we also did some work on the on natural amino acids, and we were able to introduce AZ group, PAMF. So it's a analog of phenylalanine, and we put AZ group on that, and with orthogonal system along with tRNA synthetase and also tRNAs, we were able to introduce the tag or the handle on the antibody, and we add the DBCL group along with MMAF, and we were able to get the conjugated ADCs. But I want to tell you the efficiency of the system is really low. So we are still optimizing the extraction of the enzymes, try to make the system more efficient. Okay, and we have been doing some cool stuff. So I told you that this is a branch linker system. To put it short, we were able to get the branch linker two monomer groups onto the hinge region, only the hinge region. But I have been asked that which hinge disulfide bond that has been linked to, but I think it's very difficult to assess that. But along with LC mass reducing and non-reducing, we were able to clearly identify the linker is on the hinge region, but not on the disulfide bond between light chain and heavy chain. So that's work on the monoclonal antibodies, because we are seeing a trend in the industry that actually it was summarized by Beacon antibody database, and this is a type of abstract at ACR 2025. But if we just saw the emergence of ADCs in the ACR 2026, if you have that summary, you will see even more bispecific ADCs and also dual payloads. So compared to 2024, the total of the ADC abstracts are decreasing, but for bispecifics, radio conjugates, and also dual payloads are rapidly increasing. And it seems that everyone is trying to do a dual payload ADC just to tackle the problem of DST's resistance. and of course some other conjugates, including small molecule drug conjugate, peptide-drug conjugate. But we are mostly working on antibodies, and we have been working with biotherapeutics quite normally because we are a CRO specialized in antibody production. So that's a quite easy job for us. And we are trying to do some work, just some showcase of a dual payload. And we are trying to tackle the problem by two-step conjugation. So this is first step to conjugate the branch linker to the Q295 sites I just mentioned, and then conjugate the MMF onto that linker. So in total, we have 4 MMF per molecule, and we use the cysteine conjugation to do DAR8 conjugation of the XDCs. So in that way, we have 2 different payloads, 4 plus 8. And so this one is quite interesting, I think. We are trying to just do a very quick proof of concept, because we know fiber exchange is a good system for early screening, but we want to check whether that can be used for antibody conjugation. And so we try to use Darfo method to conjugate the payload to one payload to one antibody. So probably DSD to zalutumab on the cysteines of heavy chain and light chain, and MME to another antibody, patritumab, also to the light chain heavy chain. So the hinge region is untouched, and then we are trying to do a 5' exchange, and of course we will do some mutations on the C3 parts of 2 antibodies, and then we do the change so for the end products, we can only use the mass spec to identify the DAR value, but at the end we were able to see that we can get a DAR4 molecule as we expect to do. Yeah. And we have been working with more complicated molecules. This one is so peptides and oligos has been conjugated to the antibodies. I think for the case of peptide, the GLP-1 analog peptides has been conjugated to the GLP agonist antibodies. And for AOCs, then you have antibodies of transferrin receptor against the transferrin receptors and the different oligos conjugated to antibodies. So we have done such project to conjugate the peptide to the antibodies, and the peptide is quite neat because you can see the dramatic shift on just on the SDS page, the naked antibodies and the antibody peptide conjugate in the reducing and non-reducing gel, and we use the engineering system approach to get the products, and also we have done some antibody oligo conjugate projects to have a different equivalent of oligos conjugate to the antibodies, DAR1 part and also DAR2 part. All this is also using the engineering system approach, and we are welcoming a lot of radio conjugate projects, but of course we don't have a radio facility, so we are just conjugating the chelator. So this is a typical format of the chelator DOTA group, but of course you you have the linker on some part of the carboxyl group to attach to like a PEG group, and then you have a maleimide group attached to the antibody. But we were able to get different DAR values coming from the chellator, DAR 6 and around DAR 2 value for different purpose and different metal ions. Of course, we will hand the conjugate molecule to our customers for them to add the radioactive ions, not us. And we did some new stuff. And in vivo CAR T, everyone is talking about in vivo CAR T, and it's a very rapid alternative to ex vivo CAR T. It takes a lot of time, probably more than 22 days, to make the CAR T cells coming from the body of the patients. But for the in vivo CAR T, you can use different formats, lipid nanoparticles, antivirus, and other parts. But I think the major ones are antivirus and lipid nanoparticles. And of course, for antivirus or other virus, you can fuse the antigen target region to the package of the virus. But for nanoparticle, we will need a conjugate method. So we have developed that method to conjugate the VHH using maleimide group. So we add a maleimide group to one component of the lipid nanoparticle, and then we add the nanobodies on that, and they can see on the SS page, Western blotting, there's a clear shift between the naked antibodies and also the LNPs. And we have seen the radium or radius shift from unconjugated LNP to conjugated LNPs. So we have a shift of around 12 nanometers, and you can see the distribution is quite uniform. And we have done some quick functional assays and tried to see whether the conjugated lipid nanoparticle combined to the CD8 and CD8 positive cells. We use overexpression CHOK1 cell, and we were able to see the shift on the FACS figure. So as Quentin said, we want to share the trend of industry and our experience. So we have done quite a different set of instruments, including different payloads, toxins, antisense oligos, siRNAs, peptide and AMPs, and the different antibodies, monoclonal antibodies, bispecifics, fusion proteins, and nanobodies, of course. We have been working with normal cystic conjugations, but recently we have seen more MTD conjugations and engineered cysteines, bridge cysteines, and even multi-step conjugations. And I think around 1 year ago, we were working with the DAR 8 projects mostly, but right now the distribution of DAR value are quite diverse, and from 1 to 8 normally. Even for toxin molecules, I think for high potent toxins, people tend to work on low DAR value, and we have expertise in most of chemistry conjugation methods, including maleimide, click chemistry, and other electrophile linker to cysteine. And we have done quite a a lot lysine conjugation, especially for labeling purpose. So we have been working with FITC molecules and also biotics. So that's also a conjugation, but for different purposes. And we have been doing very early screening projects like a milligram or hundreds of microgram to later late stage projects, gram level projects. And we are equipped with a lot of different QC methods. For example, RP-HPLC, HIC, AX, LC-MS, and DLS. And we are expanding our toolkits. So so I think I have been convincing you that we have a lot of expertise on the conjugate parts besides the antibody production expertise. So we can do bispecifics, dual payloads, antibody oligoconjugates, and also LNPs. So I want to pause here, and Christine, probably you want to release the poll questions.
Absolutely. Thank you so much for your presentation. So I will also put the poll questions in the chat, but the first one, so which area of ADC/XDC development is most critical for your work right now for attendees? So we have target selection, antibody discovery, payload and linker design, conjugation strategies, analytical characterization, or manufacturing a scale up. And we would love some audience participation if you want to either comment below on the chat that I put in or just add a comment to the chat separately. And while we're waiting for those responses, I will add the other ones. Okay, audience, we really love your participation, but if you're feeling a little shy, we'll also share these poll questions afterwards, maybe follow up with you all. I've added the other questions to the chat as well. Okay, well, while we wait, Dr. Wu, I do have a couple of questions for you.
Well, I don't know if I pause here because I have a couple of slides to
Sure,
introduce the company.
sure.
You want to ask the question first?
No. Go ahead. You can introduce the company and hopefully we'll get a
Okay.
couple of responses.
Oh, that's good. That would be great. So we are Biointron. We established in 2012, and we have a production site in Jiangsu, and also R&D and shipping site in Shanghai, China. And for US, we have a business shipping and marketing in New Jersey, and we are opening a site, the business site in Boston this year. And we have a logistics center in Europe for shipping purpose. We have more than 600 employees, and we have been doing quite a lot of discovery projects, 120, more than 120 per year, and we have more than 100,000 per year antibody delivered, including monoclonal antibodies and also bispecifics. We are on platforms including Science Exchange and also Scientist.com. and we were certificate by ISO 9001, and also cyber YDs. That's a cybersecurity certification, and also China Quality Mark certification. And we are building our service around antibody discovery, all enabled by our famous high-throughput antibody production platform. We have service including antibody discovery, alpaca VHH discovery, and also single B cell technology, and also antibody optimization, including unbiased affinity maturation and AI-assisted humanization designs. And we have been doing quite different formats of biospecifics. We have done 260 different bispecific formats, and also antibody-drug conjugates I just introduced today. I think last time we introduced our developability evaluation platform, including thermal stability, hydrophobicity, non-specific binding, and we have cell line can be licensed, that we licensed from ECACC, and we have done a lot of gram-level antibody production for pre-tox purpose. This is our alpaca farm. We have self-owned breeding base in China, and we have more than 300 alpacas. Probably we will get more this year because we are getting a lot of projects for nanobodies. And we have traceable health and immunization records and approved by Sanofi's animal welfare assessment. We have quite a large capacity facilities. We have two buildings with 10,000 square meters in use, and probably one other will be expanded in late June or early July this year. We have clean room, molecular lab room. We do all the gene synthesis and oligo synthesis by ourselves, ACTAs and shakers and bioreactors. And we have put a lot of focus on the automation. Most of our procedures are automated, including gene synthesis, plasmid prep, cell dispensing, and transfection, and protein A purifications. And this is just show one lineup. It's for plasmid prep, and also for aliquoting and labeling. We are doing it automatically because we are able to handle 3,000 samples per day, and with automation, that will be impossible to deliver such numbers of projects to our customers. So that'll be the end, Christine. You can ask the questions.
Thank you so much, Dr. Wu. Seems like the audience is feeling a little shy, but we'd love to hear any responses to the poll questions, as well as any questions that you might have for Dr. Wu. In the meantime, we've seen the field evolve from traditional ADCs toward broader XDC modalities. What do you think the biggest drivers behind that shift are?
So that's a question for me, right?
Mhm.
Okay. I think looking back at the history, the field, every field has a downturn and an upturn. And I think ADCs the field of ADC has a downturn about 10 years. And since the emergence of the DS 8201, sorry, developed by Datchenko, and the efficacy of that therapeutics is amazing. So everyone is mimicking that design of that ADCs, and that way involved the field and other modality, including RDCs and AOCs, borrowed the design of ADCs, successful ADCs. And of course, the drug is quite successful, but the resistance of DSD is real. So that leads a lot of groups to work on dual payload ADCs to tackle the resistance problem. Yeah.
Thank you so much. Then a question about the payloads and linkers and chemistry. Which linker or conjugation innovations do you think are still underappreciated in the field?
Well, that's a very good question. Uh, I just saw a new review. Uh, so uh, I think before everybody was thinking that if we have a fully perfectly designed molecule, we'll have very good clinical efficacy. But in the field of ADC, it doesn't seem that way because uh, if you have very stable linker, you might have a very strong on-target toxicity. But if you have a very unstable linker, you might have very strong off-target toxicity. So I think that's you need some balance. But uh, uh, in one particular setup, uh, I think the reverse microaddition problem by melamine group is very significant. So probably I don't think there are I don't know, but in my sense, probably there can be more group working on the ring opening mechanism of the linker just to make the linker more stable, but not that stable. So there is truly a balance of the instability and stability of the linkers. Yeah.
Thank you. Now, one about AOCs, oligonucleotide delivery and emerging platforms. So, antibody-oligonucleotide conjugates are becoming increasingly interesting for targeted delivery. What technical hurdles still need to be solved for a broader adoption?
Yeah, that's a very good question. Uh, I think we have seen a lot of good uh, clinical results for the muscle disease, right? And uh, I don't think it's really clear that the blood brain barrier problem has been solved completely. So I think we still need to wait on a very good clinical outcome, like the Daiichi's DST platform, and everybody will know which design will give you the best results on the brain disease by AOCs. But again, uh, the the complicated nature of conjugates and also the nature of the antibodies will make the prediction very difficult because before people are thinking ADCs are quite precise missiles that will target the tumors specifically, but that's not the case, right? The antibody is circulating in the body, not just on the tumor. So again, I think you might need a very good clinical results, and then you will know the truth about this field. Yeah.
Of course. And then from Biointron's perspective and your own personal perspective as a CRO and CDMO partner, how would you say the client requests have changed in the past 2 to 3 years, given your experience?
Uh, Actually, we're not a CDMO, but in the early discovery phase, I think they are working on more complicated molecules. I think if working with toxins, nobody is working on monoclonal antibodies anymore. Everyone is working on bispecifics, even trispecifics, and a lot of guys are working on AOCs. So and also radiotherapies. Because I think if you are thinking about uh, monoclonal antibodies with toxins, the field it was already ruled by uh, the likes of the XDs. So you have to be differentiated from others. Yeah.
That makes a lot of sense. And to be clear, I wasn't saying you are a CDMO. I was saying
Yeah.
as a partner
Yeah.
organization to CDMOs.
Yeah, of course. Yeah.
Yes. Okay, just one more question, or maybe two more for you.
Yeah.
Um, if you were advising early stage like an early stage biotech that was entering the XDC space today, what would you tell them to focus first?
Well,
I know that
that's
may be
re-
a broad
that's
question,
really
but
yeah, that's really difficult. Yeah, I just saw a review on the unmet medical needs of the ADC field, such as small cell lung cancer and gastric cancer or something like that. But they are all really difficult indications, right? Or pancreatic cancers, and nobody knows what will be a good target or what will be a good combination. So I think in the field of ADCs, you have a lot of trials and errors. I would suggest if you have a resource, you do the clinical really quick and uh, and try to make sense of the results. Otherwise, it's quite difficult to design the molecule uh, and suppose that it will work, because there will be a translation gap between cell and animal, and another translation gap between animal and human. Yeah.
and then we do have a question from the chat now. This person, Ivy Lau, is wondering if you have thoughts about what Biointron's outstanding advantages on in vivo CRT are. CART. The
CAR T.
question is in the
Yeah.
chat.
Yeah. CAR T. We I think we have a lot of experience in nanobodies. You know, uh, if you are thinking about in vivo CAR Ts, you might need nanobodies, because nanobody is very stable, or at least you can choose a very stable nanobody, uh, and they have great developabilities, and they can be used on antivirus, it can be used on AAVs, it can be used on lipid nanoparticle. Yeah, I think that would be a very good big advantage of our company. Yeah.
Thank you so much, Ivy, for your question, and thank you, Dr. Wu, for your response and answer. And I think that that is all of the questions, unless anyone else from the audience has any last minute ponderings. Give it a second. Okay, well, thank you again for this wonderful presentation, Dr. Wu. The on-demand
Thank you,
version of this
Christine.
webinar will be available very soon, and a link
Okay.
will be sent via email to everyone who registered. And feel free to watch this or any of our other on-demand webinars when it's convenient for you on our YouTube channel. Thank you, everyone, again. Have a great rest of your day.
Thank you.